Hello and very warm welcome. Dear students, today I will be talking about the branching pattern in plants and various types of branching. We will be discussing the monopodial and sympodial branching. We will also discuss the concept of canopy architecture. Plant branching pattern depends on nature and spatial arrangement of each of plant part, that is metamers or growth units. At any given time, it is the expression of an equilibrium between the endogenous genetic controlled growth processes and exogenous stimulations exerted by the nutrient supply and the microenvironmental climate, as well as the competition or cooperation from population or community. From botanical perspective, this expression can be viewed as the result of the repetition of elementary botanical entities through three main and fundamental morphogenetic processes of growth, branching and reiteration. Repetition of these entities induces gradual or abrupt changes reflecting different stages of differentiation in meristems, which are ordered in time and correspond to the notion of physiological age of meristems. Due to both endogenous control and exogenous effects, the development of meristem leads to some basic branching patterns that make the whole plant to exhibit complex structures. Although the architecture of some vascular plants consists only of a single vegetative axis during their whole lifespan, most display a more complex architecture consisting of several axes, one derived from another by a repetitive process known as branching. Now, let us discuss various branching patterns found in different plant species. First, let us try to differentiate between terminal and lateral branching. As you know, an apical meristem or an initial apical cell can directly split and give rise to two or more new sibling axes, which results in dichotomy or polytomy respectively. Frequently encountered in ferns and mosses, this terminal branching is rarely expressed in angiosperms. It is important to mention that this phenomenon has not been encountered within gymnosperms. In other cases, the branching process relies on the delimitation of a zone of embryonic cells just aside the initiated leaf, that is the axillary meristem. This lateral branching process is the most common among angiosperms and gymnosperms. The resulting lateral branch is characterized by the presence of one or more prophylls, which are the first foliar organs of the lateral axis. In some plants, there are at a single leaf axil more than one axillary meristem, which are then called supernumerary or accessory buds. In this case, each individual lateral meristem may be identified by the position of its prophylls, which also allows the distinction between the situation and the development of condensed and more complex lateral branching system, where second order lateral buds may be present in the axil of main lateral buds prophylls. Now, let us discuss immediate versus delayed branching. Once initiated, an axillary meristem may remain dormant or can develop into a lateral axis. A lateral axis may elongate immediately after lateral meristem initiation or after a phase during which the lateral meristem remains inactive and very often protected in a lateral bud. These branching patterns are referred to respectively as immediate or delayed branching. The terms immediate versus delayed branching should be preferred to the more traditional synaptic versus prolaptic branching used by Hale in 1978. because of etymological and historical reasons, which have rendered the later two terms ambiguous. Morphologically, these two branching patterns may generally be identified a posteriori by observation of the base of the lateral axis. Irrespective of the delay length, delayed branches present very short internodes and one or several cataphyls in their proximal portion close to the point of insertion. Thorough observation of the proximal part of lateral axis and a periodic observation and measurement of lateral meristem or bud size normally leaves no doubt as to the immediate or delayed nature of branching. In some cases, however, because lateral resting buds may be hidden during the resting phase in the axils of persistent basal foliar organs of the terminal bud, or because lateral axis in an embryonic stage may already exist inside a resting bud, the interpretation may be complicated or may lead to misuse of the terminology. Finally, in delayed branching, it has been shown that the duration of the delay may be of several weeks to a year and even several years. Now, let us try to differentiate between monopodial and sympodial branching. Depending on the indeterminate or determinate growth pattern of an axis, its branching pattern may respectively be monopodial or sympodial. In the latter case, 1, 2 or more branches may develop after the death, abscission, abortion or transformation of the apex, and the resulting sympodial branching pattern may be qualified respectively as mono, di or polychasial. Thus, in monopodial growth, the stem is constructed of a single straight shoot bearing side branches with a single axis, that is monopodium, developing from a single apical bud that continues to grow continuously or periodically. On the other hand, the sympodial plants are the truly modular plants, with the stem consisting of a series of modules stacked one on top of the other, with each terminal apical bud ending in a flower at the some stage in growth, and the main stem or branch continuing its growth with the extension of the previous modules or axillary buds, forming a new module in which again the apical bud ends as a flower, whilst the axillary bud continues growth. In plant architecture, the concept of module was defined for the first time by Prevost in 1967 in her study of tropical Apocynaceae, and then applied to a leafy axis in which the entire sequence of differentiation is carried out from the initiation of the meristem that builds up the axis to the sexual differentiation of its apex. Because of the various causes of determinate growth, this definition is however restrictive, and the term module increasingly refers to the portion of an axis edified by a single terminal meristem, which corresponds also to a sympodial unit as used by Bell in 1991. Involving one or more lateral axes, a sympodial branching system is very often three-dimensional. In some cases, however, a sympod may imply a linear succession of modules or sympodial units forming a so-called cynomonopodium, as termed by German morphologists. The resulting rectilinear structure mimics an axis edified by a single meristem with indeterminate growth, and if reproduced on all axes may give rise to a totally sympodial branching system that resembles a monopodial one. As a consequence, a rectilinear system may thus be composed of a succession of metamers or growth units or annual shoots, all produced by a single meristem or by a linear succession of sympodial units or modules. Now, let us discuss branched system and branching order. The description of a branched system implies the use of a precise topological terminology. In plant architecture analysis, it is usual to use ordinal numbers and to consider the main stem arising from seeds as the order 1 axis, whereas the axis it gives rise to are referred to as order 2 axis, and so on. In a sympodial system, a rigorous use of this terminology will lead to the reference of the successive sympodial units as axis orders 1, 2, 3, etc. In order to be coherent with the broad axis definition, each rectilinear succession of modules, even though not strictly edified by a single meristem, will be considered as an axis and will represent an apparent branching order. Now, let us try to differentiate between rhythmic and continuous or diffuse branching. Defined and used for the first time for plant architecture description, these terms contribute to the characterization and definition of architectural modules and take into account the topological distribution of sibling axes on a parent axis. Depending on whether all the auxiliary meristems of a stem develop into a lateral axis or whether lateral axes are grouped as distinct tiers with an obvious regular alternation of a succession of unbranched and branched nodes on the parent stem, branching is respectively referred to as continuous or rhythmic. In some cases, neither all nodes of a parent axis are associated with a lateral axis, nor is there an obvious regular distribution of branches in tiers, and the branching pattern is then called diffuse. As revealed in cupressaceae by qualitative observations and in recent years by sophisticated mathematical methods, a diffuse branching pattern may not mean an unorganized distribution of sibling shoots on a parent shoot, but may indicate a predictable, precise and subtle branching organization. Now, let us try to differentiate between acrotonic and mesotonic or basotonic branching. In order to describe the positional preferential development of a lateral axis on a vertical parent axis or shoot, Troll in 1937 and Raw in 1939 distinguished three modalities called acrotony, mesotony and basotony. Acrotony is the prevalent development of lateral axis in the distal part of parent axis or shoot, and depending on whether branching is monopodial or sympodial, Rah and Champagnath termed it respectively primary or secondary acrotony. In the initial definition of acrotony, the parent axis was always longer than the sibling ones, but Bell in 1991 used the term acrotonic branching for the distal position of the largest lateral branch. independent of its size relative to parent stem. Basitony, on the other hand, was at first considered as the preferential development of lateral axis in the basal part of a vertical stem. Bell in 1991 used the term basitonic branching when the proximal branches grow larger than the distal ones. Finally, mesotony is used for a privileged development of branches in the median part of a shoot or axis. Clearly included in the definition or implicitly shown in the illustrations, the topological lateral arrangement of branches along the parent axis is often associated with an increasing or decreasing gradient in length and or vigor of the branches. As discussed by Caraglio and Barthelemy in 1997, these 2 points must nevertheless be considered separately, as the diversity of their expression in plants shows that there is neither automatic nor direct correlation between the privileged position and the relative vigor of lateral branches. As all kinds of combinations may be found in the plant kingdom, we would prefer the terms acrotony, basitony and mesotony to be used only in reference to the privileged location of branches on a parent shoot, respectively distal part, proximal part and median position, without reference to their relative vigor or length, which can be given in precise terms in addition. Acrotony or basitony are frequently considered as two fundamental phenomena underlying respectively the arborescent or bushy growth habit. Nevertheless, these authors refer mainly to the acrotonic branching of growth units or annual shoots in the arborescent case, whereas they consider the proximal branching at the base of the whole individual when considering bushy plants. As discussed by Caraglio and Barthelemy in 1997, either acrotonic or basitonic branching may characterize the growth units or shoots of either tree or bush, whereas basal sprouts may also be an adaptive strategy of some plants like trees. Now, let us discuss hypotony, epitony and amphitony. In order to describe the privileged arrangement and lateral axis on a horizontal, curved and splanded parental axis, Droll in 1937 defined three modalities for so called lateral symmetry. According to this terminology, the privileged development of branches on the upper lateral or basal portion of a parent axis is referred to respectively as epitony, amphitony or hypotony branches. Epitony is a very common process in many fruit trees and in plants belonging to such architectural models as those of Champagnat, Troll and Manginat. It is also often associated with the survival of old branches in the canopy of old trees. Hypotony is frequently marked by a privileged development of lateral axis in the curvature zone of a slanted branch, whereas amphitony is frequently on rectilinear, horizontal or slightly slanted. Hypotony and amphitony may be combined in slanted and curved branches, and their incidence in expansion of lateral branch complexes is of the utmost importance in the crown architecture of many woody plants. Finally, it is noticeable that amphitony is frequent feature in rectilinear branches, whereas epitony and hypotony are characterized by the predominant development of lateral axis on the convex side of the curved downwardly or upwardly oriented branches. As highly influenced by axis orientation, these phenomena are frequently combined with the previously described topological arrangement along axis, and these combinations have considerable relevance to bud fate according to their topological position and space orientation within a plant crown. With this, we conclude our topic plant architecture and plant branching. Hope you enjoyed the topic. Thank you.